in this study where we look at these hyperdiploid, pre and post-hyperdiploid changes, one question we had in our mind, how many of the mutations, when we do a whole genome sequencing, we do see around 7,000-8,000 mutations for a given patient at the bulk sequencing. And one of the key questions we had in our mind, how many of these mutations would be actually there before the so-called primary events happen. Hyperdiploidy and some of the translocations has always been considered as the early events and the initiating events for normal plasma cells to go to precursor conditions and then eventually become myeloma. And we wanted to see how much of the mutations were actually already there before any of these events happen. And hyperdiploidy gives us a very good model to look at it. We were able to differentiate the mutations at pre and post stage by looking at their variant allele frequency, and this basically told us or showed us that there are still 1,500 to 2,000 mutations that are already accumulated before hyperdiploidy happened. And then we have seen or we have shown before in our publications that hyperdiploidy has much less mutational load than non-hyperdiploid patients. We wanted to see if there would be a specific reason for this, and then for the hyperdiploid patients, we were able to compare their diploid genomes with the hyperdiploid genomes, and by controlling all the factors like the detection power and all additional features, we were actually seeing much less mutation accumulation after the hyperdiploid happens in these particular chromosomes. That brings us to the point that like hyperdiploid may be causing some decrease in mutation accumulation over the time. We have also looked at the mutational signatures accumulated pre and post hyperdiploid to see how the mutational processes have changed, and this also have shown that processes like APOBEC or the DNA damage is picking up after the primary events happens, and then before the primary events happens, everything seems like controlled by either time or clock-like mutations and the AID as possibly the just regular biological process of B-cell differentiation.